All right, guys, so here we go into chapter 7. I think this is the last chapter for this exam 2. This exam 2 is going to be difficult. I think some of the hardest chapters that we're covering for this exam. So chapter 5, 6, and 7, muscle, bone, and nervous system. They are probably the hardest systems to learn. So put in some extra time for this next exam and study hard and get into groups, talk to each other, help each other out. So we're going to talk about the nervous system. I'm going to get my lecture going. We're going to overview the nervous system and its organization, and then talk about neurons, the major cell type of this system. So this is our next organ system that we're going to talk about, the nervous system. So we just covered the muscle system, skeletal muscle system, and the skeletal system. And we're going to head into the nervous system and how it controls and modulates and does all kinds of crazy stuff. And it is divided into two major components: the central nervous system, made up of the brain and spinal cord, and the peripheral nervous system, which is basically everything else. If you're not the brain and spinal cord, then you're part of the peripheral nervous system. I think peripheral means away from central, right? So like your peripheral vision is what you can see off the side of your eyes. Like if you're looking forward and you move your hand next to your head, you can still see your hand moving. So the peripheral nervous system is everything out in your arms and your legs and in your trunk area, the thoracic cavity and the abdominal cavity. That's all. There are sensory neurons there, and those sensory neurons, or afferent neurons, are part of the afferent nervous system. The sensory nervous system, it's a division of the peripheral nervous system that sends information about your body or the external environment. So you're basically sending a whole bunch of information. Think about it like data. You're sending data to your central nervous system to say, here's what's happening with the internal or external environment, so that the central nervous system can integrate that information, process it, and figure out what to do about it. And that then basically takes the sensory information and sends more information by way of the motor nervous systems, the efferent division of the peripheral nervous system, which is the motor division, to cause some response and cause commands. Basically, it just commands things to happen. So it's all one big system of communication from the peripheral nervous system. The sensory division is sending information to the central nervous system to be integrated and processed. And then the central nervous system sends signals back out to the body, the rest of the body, and the peripheral nervous system by way of the motor division to coordinate the response to the original sensory information coming in. So it's all one big pathway. A sensory division is divided into or is comprised of several components. It basically brings information from receptors. And think about what a receptor would do in your body. A receptor receives information. Like there's temperature receptors that can sense changes in body temperature or external temperature, touching your skin. There's tactile or touch receptors that receive information about touch. There are blood pressure receptors that sense changes in blood pressure. There are, and those type of receptors are considered visceral sensory receptors. They are monitoring internal organs. You have, so the word visceral, this word visceral here means internal organs. So you actually have sensory receptors that are receiving information about changes that are occurring in internal organs. And those sensory receptors can send information by way of a sensory neuron to the central nervous system, the brain and the spinal cord, to say, here's what's happening with this organ. Let's keep it safe and healthy. There's somatic sensory receptors, and the somatic senses basically are the senses that are sending information to your brain about touch, body position, pressure, pain, and temperature. I mentioned temperature receptors earlier and touch receptors. These are considered somatic sensory receptors. And then there's special sensory receptors in like the eye for vision, in the ear for balance and hearing, on the tongue for taste, and in your nose, your nasal cavity for smell. So those are special sensory receptors that send information to the brain about those special senses. The motor division of the peripheral nervous system, so that was the sensory division of the peripheral nervous system. This is now the motor division. And we'll come back to more detail about all of these divisions later, but I'm introducing them now. Basically, the motor division takes the information that the central nervous system has received and integrated, and then carries information about that integration out to some effector. And an effector is something I will also refer to as a target tissue. So an effector is basically, if you think about it like one big feedback loop, the receptor is receiving information about some sensation, right? Sending that information by sensory receptors to the central nervous system to say, okay, this is what's been happening in the internal or external environment, and now we need to do something about it. Well, doing something about it is basically taking the motor neurons that are sending information to an effector, some tissue in the body, to cause a response. And there's two types of motor neurons, and there's two divisions of the motor division of the peripheral nervous system. The somatic nervous system that has neurons that innervate. If a neuron innervates a tissue, it's basically sending information to that tissue. The somatic nervous system is carrying messages to an effector, which is skeletal muscle. So think about like your bicep muscle or one of your quadricep muscles in your leg, or your tricep muscle. Any of those muscles are skeletal muscle. So the somatic nervous system is basically somatic neurons. They're motor neurons that are sending information from the central nervous system to skeletal muscle. The autonomic nervous system carries messages to pretty much every other tissue in the body. So smooth muscle, cardiac muscle, fat tissue, glands, any tissue that's not skeletal muscle is innervated by motor neurons of the autonomic nervous system. And here's a figure that shows those divisions. So here's central nervous system versus peripheral nervous system. Here's the sensory afferent division sending signals to the central nervous system for processing. And then that processed information and signals about that are sent by the motor efferent division. And if that information is headed to an effector that is skeletal muscle, it's headed there by way of somatic neurons in the somatic nervous system. Or if it's headed to every other type of gland or tissue, the heart, blood vessels, glands, fat tissue, that information is being sent along autonomic neurons in the autonomic nervous system to that tissue. The major cell type of the nervous system is a neuron. And there's some basic components of neurons that you need to understand to be able to understand how neurons communicate to one another. And one of those structures is called a dendrite. And there's a big red arrow here. Dendrites are extensions of the cell membrane. So if this here is the cell body, and here is the cell membrane, look at how that cell membrane just kind of extends out and creates these little structures here. These are all dendrites. And those dendrites have receptors on them that are receiving information. So dendrites receive a stimulus from another neuron. They are really short and highly branched, and they are all branched off of the cell body. And when they receive information, these dendrites receive stimuli from the environment or other neurons. They send that information along the cell membrane to this area here, where all that information gets summed. We'll talk about this area here shortly. So where all that stimuli information, the information about stimuli, collects right here, the information is sent along the axon to be communicated to other cells. And that axon is basically carrying information away from the cell body towards other cells. So where that axon ends, there's another junction with another neuron or another cell. We'll talk about that later. So 3 major structures in a neuron: dendrites, receiving information, receiving stimuli from either the environment or other neurons. The cell body collects that information and sums it here, where the cell body meets this long extension called the axon. And the axon then sends information, basically takes that information and allows it to be communicated to another cell. This area here that's so important is called the axon hillock. And the axon hillock is the area where the cell body here meets the axon. And that's where the information being received by dendrites is collected and processed. And that process of information can be excitatory information or inhibitory information. We'll come back and talk about what I mean by that here shortly. That excitatory information is sent along the axon to the axon terminal. Think about where the axon terminates or ends. It's called the axon terminal. And that axon terminal is so important and critical to the function of a neuron. It's basically where communication occurs between this neuron. It's called the presynaptic cell. It's before the synapse. And the synapse is basically the junction between the axon terminal and the next cell, whatever that cell might be. It's called the postsynaptic cell. It may be a neuron, it could be a muscle fiber or cell, and it could be a glandular cell, it could be any other cell in the body neurons are able to communicate to. So the presynaptic cell before the synapse is this neuron. And the part of the synapse that this presynaptic cell has is the axon terminal. So the axon terminal is the portion of the presynaptic cell that releases and sends information across the synapse to the postsynaptic cell on the membrane there. And if it's a neuron, that postsynaptic cell, the membrane of it may be the membrane of a dendrite. So this synapse here is a junction from one neuron to this postsynaptic cell, where that may happen here as well. So if there's another cell that was coming in and sending information to this dendrite where there's a receptor, there could be a synapse here as well. So to communicate from one neuron to another cell, there has to be a synapse. The synapse is basically the point of communication between the neuron and the next cell. So I've added some slides in here for review, just as a kind of checkpoint along the way. Where you should be able to name structural components of a typical neuron and what the functions of each of those components is. So you'll see some checkpoints that sometimes I'll stay on as we get used to this in the new chapters, and sometimes I won't. I'll just kind of breeze past them, but they're there for you to study. So neurons are the major cell type of the nervous system, but there are also other cells that help neurons do their job and stay healthy along the way. So neuroglia of the central nervous system, there are 4 types of cells that are collectively called neuroglia or glial cells. And they do not function in communication like neurons do, but they support and protect neurons while the neurons of the nervous system are doing their job. And there's types of neuroglia or glial cells in the central nervous system, and cells that are similar to those cells of the peripheral nervous system that have a similar job, but they're not located in the central nervous system. They're located in the peripheral nervous system. Those 4 glial cells of the central nervous system are called astrocytes, ependymal cells, microglia, and oligodendrocytes. We're going to talk about each of them now. So ependymal cells, and on this slide, you can see there's here is a layer of ependymal cells. Those cells form a lining between that separates basically the brain over here and a cavity here. And these ependymal cells basically create these fluid-filled passageways in the spinal cord and the brain. And the fluid within those passageways is called cerebrospinal fluid. So think cerebro is brain, spinal is the spine, or basically the spine. There's cavities and passageways in the brain and in the spine where these ependymal cells produce cerebrospinal fluid, or CSF. We'll talk about the function of CSF later. So for now, the job of the ependymal cells is to produce CSF, cerebrospinal fluid. And CSF, this fluid, is a lot like plasma in blood, the fluid component of blood, but it's a little bit cleaner. It actually gets filtered as it makes its way from the blood. It's basically fluid that's taken from the blood without the blood cells and passed through the ependymal cells and then secreted into these spaces or passageways. Microglia are little garbage disposals. So there's a cell called a microglia right here. They remove cellular debris and waste products and pathogens from the central nervous system so that those pathogens and waste products and debris don't cause problems, don't cause infection or anything like that. So they're basically like little garbage disposals running around, chewing things, eating things up so that they don't cause problems. So they're similar to macrophages, and macrophages are able to phagocytose, or basically eat up things that aren't supposed to be there. Astrocytes are these large cells here. Astrocytes maintain something called the blood-brain barrier. Think about what those three words mean to you. The blood-brain barrier. It's a barrier between the blood and the brain. Pretty simple, right? Basically, it just provides structural support. And these astrocytes are like a bouncer. They regulate what's allowed into or out of the brain in exchange between the brain and blood. Because there's a lot of things in blood that can be carried and transported that shouldn't make their way into the brain, because that could cause problems. Certain drugs and toxins, waste products. There's a lot of things a lot of chemicals or substances that if they were to get to the brain or into the brain and the spaces located there, they may cause infection or problems. So the astrocytes are maintaining this barrier from the blood to the brain so that only certain things can get through. And those things that can get through are important to the function of the brain. Lastly, oligodendrocytes, you see them here. Oligodendrocytes, there's the nucleus of an oligodendrocyte, and then look at how this cell wraps around this long structure here, the axon. So back here is the cell body, here's some dendrites. Right here would be the axon terminal of one neuron and the cell body of the next neuron. So this would be this whole structure here, this area is a synapse. There's the axon hillock of a neuron and the axon. So look at what this oligodendrocyte is doing. It's wrapping part of its membrane around the axon. That part of the membrane that wraps around the axon is called myelin. And myelin is this really important substance that surrounds axons in both the central and peripheral nervous system. But in the central nervous system, oligodendrocytes produce myelin. In the peripheral nervous system, another cell does that. But this myelin insulates axons so that the electrical signals sent by these axons conduct or propagate or move faster along the axon, as opposed to axons that don't have myelin, because not all axons do. Axons that don't have myelin, the electrical signals are conducted along the axon a little bit slower. So myelin is this sheet of tissue that wraps around the axon. It's produced by oligodendrocytes, and it can help protect but also insulate the axon to speed up how fast electrical signals are sent from the axon hillock to the axon terminal. This myelin sheath, I got a little ahead of myself on the previous slide, so it kind of describes that here. Myelinated axons, myelin again is produced by oligodendrocytes, wrap around the axon, and where myelin is located and wrapped around the axon is called an internode, between the nodes. Gaps between the internodes are called nodes. So take a look back at this figure right here. There it is. There's an internode, so that the part of the axon that's covered by myelin is called the internode. But there's a small gap between one internode, the area of the axon covered by myelin, and the next internode covered by myelin. That's called a node, or a node of Ranvier. So that node is an area of the axon where there is no myelin, and that's really important, and we'll talk about why it's really important later. So there's gaps. So that's actually labeled right here. There's a node of Ranvier, or a node, and here's a node. It's a gap or a space, a portion of the axon that's not covered by myelin. Most of the nervous system where there is myelin has this white sheen to it. And if you've ever heard of the difference between white matter and gray matter in the central nervous system or peripheral, in the nervous system in general, typically where there is white matter, there are myelinated axons. Where there's gray matter, there's unmyelinated axons or cell bodies, things like that. Regions of the central nervous system where there, again, are cell bodies and unmyelinated axons, they appear more gray because there isn't the white, shiny white matter located there. And there's another review. So here's a good view of internodes. So here's where myelin is located. These little gaps of the axon where there is no myelin are nodes. And basically the electrical signal that's generated here at the axon hillock in response to the collection or summation of all the stimuli coming into dendrites, being received by dendrites, is collected here and then conducted, basically generates an electrical signal, all of that information, the sum of all of it, and is sent along the axon. And it's sent along an axon faster where there's myelin. In the peripheral nervous system, oligodendrocytes in the central nervous system produce myelin. In the peripheral nervous system, the cells that produce myelin are called Schwann cells. And the outer surface, the membrane of the Schwann cell is called the neurolemma. And basically, just like oligodendrocytes, Schwann cells in the peripheral nervous system produce and form myelin sheaths that wrap around the axons in the peripheral nervous system. So usually motor neurons in the peripheral nervous system, a lot of them have myelin, especially in the somatic nervous system, where neurons are innervating, remember, skeletal muscle. So the motor neurons of the somatic nervous system are typically covered, surrounded by a myelin sheath to create internodes. Satellite cells surround cell bodies in the peripheral nervous system. So they're similar to kind of like neuroglia. Clusters of cell bodies. So cell bodies in the peripheral nervous system cluster together, and they're surrounded and protected in those clusters. There's satellite cells there that are basically located in the same area as these cell bodies in the peripheral nervous system to help protect those cell bodies and keep them safe and healthy. Well, those clusters of cell bodies in the peripheral nervous system are called ganglia, and they're usually surrounded by some kind of tissue that helps protect them. So you don't just have cell bodies of neurons in the peripheral nervous system all over the place. They cluster together and collect together to stay safe in these structures called ganglia. We'll talk more about ganglia as we go, because there's specific locations of peripheral ganglia. In the central nervous system, a ganglia is called a nucleus. So a nucleus in the central nervous system is basically a cluster or collection of cell bodies. In the peripheral nervous system, that collection or cluster of cell bodies is called a ganglia. So similar structure, different name. Functionally, there are different classes of neurons. So there's 3 functional classes of neurons, meaning that these 3 different types of neurons have different functions. Sensory neurons are one type of neuron functionally. They're receiving information from sensory receptors and sending information from the peripheral nervous system to the central nervous system. Let me see if I have it. Yeah. So cell bodies of sensory neurons are all collected. Remember, a ganglia is a collection or a cluster of cell bodies in the peripheral nervous system. Cell bodies of these sensory neurons cluster together in a specific structure we'll talk about shortly. And basically the job of these sensory neurons, the function of these sensory neurons, is to deliver information that was received from sensory receptors in the peripheral nervous system and send that information to the central nervous system, the brain or the spinal cord, along afferent or sensory fibers, and those fibers are axons. So basically sensory receptors are activated by some change or some stimulus. And then from that point of receptor activation, from the stimulus, sensory neurons, afferent neurons, carry that information, transport that information about the activation of that receptor, about that stimulus, all the way to the central nervous system to be integrated. Interneurons basically receive sensory information from sensory neurons and can communicate between a sensory neuron and a motor neuron. So think about interneurons being the go-between, the middleman. They are a neuron that's between 2 other neurons, an interneuron between neurons, right? And they are kind of responsible for some higher functions. Memory, planning, and learning are higher functions, not just simple functions. So they can receive sensory information from a sensory neuron in the peripheral nervous system These are typically located in the central nervous system, and they can communicate to a motor neuron or another interneuron. So they are a kind of a middleman or a go-between. And then motor neurons carry information from the central nervous system where it was integrated, right? So a sensory neuron sends information to the central nervous system to be integrated. Central nervous system then integrates that information, processes it, and decides what to do about it. And then the central nervous system sends information to motor neurons to carry that information to the effectors, the end of this loop. And those effectors are either, again, target tissues like skeletal muscle, and that skeletal muscle can be under voluntary or involuntary or subconscious controls by way of somatic motor neurons. Remember, the two divisions of the motor division and the peripheral nervous system are the somatic motor nervous system or the visceral, which is autonomic nervous system. So somatic motor neurons carry information to effectors like skeletal muscle by way of peripheral nerves. Like, so think about like the sciatic nerve. If you've ever heard of the sciatic nerve, that's a peripheral nerve that's carrying motor information that was integrated. Basically, the central nervous system said, okay, motor neuron, here, take this information and carry it to these skeletal muscles. The sciatic nerve is one of the peripheral nerves that carries motor information, electrical signals, and communicates to muscles in the upper and lower leg. Visceral motor neurons carry information that was integrated from the central nervous system to other effector tissues like smooth muscles, glands, fat tissue. And they communicate with other maybe visceral motor neurons in other ganglia, so collections of cell bodies in the autonomic nervous system. That may be a little bit confusing now with regard to what that means, but we'll come back and talk about that later. And there's a figure that summarizes all of that. So you could be looking at that and maybe rewind what we just talked about and kind of go through that together. Sensory neurons here, there's interneurons between two other neurons, right? So that could be an interneuron communicating between sensory and motor, or an interneuron communicating to other, motor neurons. There's interneurons are your go-between. And then these blue here and red here would be motor neurons of the somatic nervous system or autonomic nervous system, carrying that integrated information to the effectors to cause a response. So basically these effectors are responding to an original stimulus. So here's where a stimulus starts. Sensory information about that stimulus is sent to the central nervous system, and then motor neurons take that information that has been integrated by the central nervous system, send that information to effector tissues to cause a response. So stimulus, response. And here's the whole pathway that makes its way from stimulus to response. And an example of a stimulus and response would be like, your body temperature increases. And if your body temperature increases, you get hot, right? Your body feels overheated. The central nervous system is receiving information about that change in body temperature and sends information to effector tissues like your skin and muscle in certain areas so that you sweat to cool back down, right? That would be a stimulus, body gets too hot, and response, you sweat to cool down. And there's a review. All of that communication occurs by way of changes in something called membrane potential. So this is a sticky topic, but we're going to talk about this for the rest of this first lecture. Membrane potential is basically boils down to charge and a separation of charge. And that separation of charge is separated. So think about measuring charge in like millivolts or volts, right? So you can measure charge in millivolts, and that's what we measure charge in the inside of our cells. The inside of the cell has a different charge than the outside of the cell because of the cell membrane. So there's basically an unequal distribution of charge, and think about what contributes to charge. Positive ions, negative ions. So a positive and negative, so thinking about changes in charge. If you wanted to increase and have the inside of the cell become a more positive charge, then you may add positive ions to the inside of the cell. But what's separating and creating this unequal distribution is the cell membrane. So the cell membrane creates a barrier, and it isolates the inside of the cell from the outside of the cell. And remember that the cell membrane is Semi-permeable. It allows certain things to cross through the membrane from one side to the other, inside to outside or outside to inside, and other things it won't. It selects, so it's selectively permeable to certain ions, certain substances in the body. So this cell membrane is so important. It creates this distribution or this separation of charge from the inside of the cell to the outside of the cell. And that's basically what membrane potential boils down to. It's a separation of charge where the inside of the cell is more negative than the outside of the cell, the whole environment. So think about the fluids and the organelles and the ions and proteins floating around inside a cell versus outside the cell where there's other fluid and other tissues and other cells and all kinds of things happen out there. Well, in the extracellular fluid, there's more of a positive charge because there's more positive ions and fewer negative ions, whereas inside the cell, within the membrane, encased inside that membrane, there's more negatively charged ions than positively charged ions. So the inside of the cell is more negative than the outside of the cell. And what allows that to stay in that state in a resting cell is the membrane, because it's isolating the inside from the outside. Basically, neurons in your body and the way that the nervous system communicates from one neuron or a receptor to a neuron or a neuron to another neuron or a neuron to an effector tissue like a muscle cell or a gland cell is by way of changes in membrane potential. So membrane potential in a resting cell is basically the charge is measured as the charge inside the cell that tends to be more negative than the outside of the cell. And when that cell membrane at rest changes, that's basically what generates an electrical signal in our body. And that electrical signal is called an action potential. So we're going to talk about that as we go. So here's a distribution of charge. This figure you may see in your book, and I added some things to it to kind of help make some sense of it. In a resting cell, so the cell is not doing anything. It's not sending an electrical signal. It's not doing anything. So this is a resting neuron, call it. Here's the plasma membrane. Remember, the membrane is this barrier that separates and isolates the inside of the cell here, this kind of peachish color, and from the outside of the cell, this darker blue color. This is extracellular fluid, intracellular fluid, or cytosol. Those are the same. Intracellular fluid and cytosol are the same thing. So in the extracellular fluid, because the membrane separates the inside from the outside of the cell, there's a much higher concentration of sodium ions than there is inside the cell, which is why sodium is huge out here and sodium is small here. So there's tons of sodium ions outside the cell and just a few sodium ions inside the cell. Well, that's the opposite for potassium. So potassium, the symbol is K, sodium is Na. So this potassium ion is bigger inside the cell than it is outside the cell because the concentration of potassium inside the cell is much higher in than it is outside the cell. And that separation and those concentrations are allowed to stay low out and high in, or low in and high out, because the plasma membrane is there and it's isolating and separating those two fluids, and the membrane doesn't allow sodium in a resting cell to move across this membrane at all. In a resting cell, potassium can kind of leak out a little bit over time. So as potassium kind of leaks out over here, this concentration gradient is maintained by other things that kind of pump potassium back into the cell. So the plasma membrane in a resting cell is just a little bit leaky, a tiny bit permeable to potassium, but impermeable to sodium. So for ions, because the membrane is selectively permeable and it only allows certain things through, something like sodium or potassium that are in a resting cell pretty impermeable, they don't move very easily across the cell membrane. They have to have a channel to get across the membrane. So there's membrane-bound proteins. This protein happens to be a sodium channel. And if it's a channel, it's basically a pore or a tunnel through the cell membrane that allows sodium and only sodium to diffuse through. So remember, sodium is high in concentration outside the cell in a resting cell and really low in concentration inside the cell in a resting cell. If you happen to open a tunnel in the cell membrane that allows sodium to diffuse through, it'll diffuse down its concentration gradient from where sodium is high outside the cell to where sodium is low inside the cell. And that's pretty much the only way, one of the only ways that sodium can get across the membrane is through a channel. And these channels can open and close. So they don't always stay open to allow sodium to move however it wants to. As otherwise, if it was always open, the concentration gradient of sodium would basically equalize across the membrane from in to out, but it doesn't. So these sodium ion channels are closed in a resting cell, and they can be opened and closed in response to a couple of different types of stimuli. So there basically has to be a stimulus to open this channel or close it. We'll talk about those stimuli as we go. Same thing goes for the potassium channel. And in these figures, potassium is always going to be this, the potassium ion is always this peach color, and potassium channels that potassium can move through is the same peach color. Sodium always has this kind of purplish blue color to it, the ions and the channels it's able to move through. So we'll always follow those colors along the way. So membrane-bound channels allow some sodium and potassium. Some are always open. Some potassium channels are always open, but most of the time, sodium channels are always closed. But they can open in response to the binding of a neurotransmitter. So a neurotransmitter, look at that word, neurotransmitter. It's a chemical signal that neurons can release. And those neurotransmitters can bind to channels like this. There's actually a site or a receptor spot on there that neurotransmitters can bind to. And if a neurotransmitter binds to this channel, it tells that channel to open and allow sodium to diffuse through. So some of these channels open and close in response to the binding of a neurotransmitter to that channel. Other channels open and close in response to changes in membrane potential. So remember, the inside of the cell in a resting cell is negative compared to the outside of the cell. So if that membrane potential changes from that negative value to some more positive value or some more negative value, that membrane potential, that change in membrane potential can stimulate the opening of some of these channels too, which would allow those ions to move across the membrane. Some transporters are also located in the membrane. So there's an example of a transporter. It's called the sodium-potassium exchange pump. And it uses our energy molecule ATP. So it's not necessarily a channel, it's a pump. And it will pump sodium ions from the outside of the cell to the inside of the cell. Excuse me, I flip-flopped that. From the inside of the cell, it'll pump sodium out. And these are basically pumping these ions against their concentration gradient. So where sodium is typically high outside the cell, this pump is basically using the energy donated from ATP to pump sodium out of the cell and then pump potassium into the cell. And basically what changes in membrane potential boil down to, and an action potential, this electrical signal that we're going to talk about, simplifies to, is when a neurotransmitter or something changes the open state of this channel and allows sodium to diffuse in down its concentration gradient, that sodium that rushes into the cell down its concentration gradient is pumped back out of the cell by the sodium-potassium pump. And then when potassium is allowed to diffuse out of the cell through this channel, it gets pumped back in up the concentration gradient by the sodium-potassium exchange. So basically the sodium-potassium pump is exchanging sodium ions for potassium ions as they're allowed to diffuse in or out of the cell. So it's basically just using this pump that's also in the cell membrane to just reestablish the concentration gradients out of the cell and in the cell between sodium and potassium. So we just talked about a couple of changes in membrane potential, but changes in membrane potential the cause of that or the response to that are basically threefold. A change in membrane potential can trigger a muscle to contract. So if there's a communication between a neuron and a muscle cell, changes in membrane potential in the neuron can cause a muscle to contract or a gland to secrete whatever it secretes, like a sweat gland or a gland in the digestive system to secrete digestive digestive enzymes. Or one neuron can communicate to the next neuron by changing its membrane potential and sending that signal to the next neuron. It's crazy, right? So changes in membrane potential, I've been mentioning this resting cell, this resting potential, and it's negative. A resting membrane potential is negative, meaning that the inside of the cell has a more negative environment than the outside of the cell. So there's fewer positive ions inside the cell, and there's more negative ions inside the cell that create this charge. It's a negative charge called the resting membrane potential. So remember, membrane potential is basically just a charge, and that charge at rest is negative. A change in resting membrane potential can be a local small change called a graded potential. And basically a stimulus has to cause a change in resting membrane potential. So a resting membrane potential can change to increase to a more positive value, or it can decrease from the negative value at rest to an even more negative value. Those small changes are called graded potentials, a small increase or a small decrease in resting potential. And if a graded potential becomes big enough, so if an increase in membrane potential, this small increase called a graded potential increases membrane potential to some more positive value, you can trigger what's called an action potential. And an action potential is our major electrical signal in our body. And that action potential is basically what axons in neurons use to communicate from one axon of one neuron to the next neuron, or to a muscle, or to a gland. So our action potential is this electrical signal that we use to communicate from our nervous system to other cells by way of a synapse. So remember, back to the neuron, the picture of the figure of the neuron, where the axon ends at the axon terminal. So where the axon ends at an axon terminal, that axon terminal is one component of a synapse. And a synapse is basically the point of communication between the neuron and the next cell. So in that neuron, the axon terminal is called the presynaptic cell, remember, before the synapse. And a synapse is basically a space. We'll talk about that here shortly. That presynaptic cell, the neuron, the axon terminal of the neuron, releases neurotransmitter, that chemical signal. Remember, we said neurotransmitters are chemical signals that neurons release. That neurotransmitter binds to a receptor on the postsynaptic cell after the synapse. That could be another neuron. It could be a muscle cell membrane. It could be a gland cell membrane. And that binding of neurotransmitter to the receptor on the postsynaptic cell then changes the membrane permeability to ions of that next cell. And that can then change resting membrane potential of the postsynaptic cell and generate another response in the postsynaptic cell. So thousands of axon terminals can communicate with one neuron in the cell body of one neuron by way of, again, remember, dendrites are receiving information, are receiving neurotransmitters. So basically, when we said earlier, dendrites are basically receiving stimuli from other neurons or other cells or other environment, they're receiving information by way of neurotransmitter or changes in membrane potential, right? So resting membrane potential changes, and that is a signal to the cell that a stimulus has occurred. So here's where this happens. This cell here is our neuron, remember? So this is the axon terminal. So here is a synapse, here's a synapse, here's a synapse. Those are structural, basically just structures. A synapse is made up of three components. The presynaptic cell, which in this figure is the axon terminal. The postsynaptic cell, which is the next cell, the membrane of the next cell. And then the gap or the space between the two, it's called the synaptic cleft. So one whole synapse is made up of one presynaptic cell, a synaptic cleft, the space between the presynaptic cell and the postsynaptic cell after the synapse. So resting membrane potential, remember, it's negative. Inside the cell here is more negative than everything around it. A change in membrane potential occurs from rest because the dendrites here have received stimuli, and that could be excitatory stimuli or inhibitory stimuli. And those stimuli, each one of them changes membrane potential ever so slightly. So it can increase or decrease membrane potential because of a stimulus in these dendrites. Well, all of those stimuli excitatory or inhibitory, collect, they sum here at the axon hillock. And that there's graded potentials that occur here. And graded potentials can be an excitation, an increase in membrane potential to some more positive value, or a decrease in membrane potential to a more negative value. Well, the sum of all of those stimuli, those sum here to one graded potential. And if the graded potential is excitatory enough, we'll talk about what that means here shortly, that graded potential can stimulate an action potential to occur here. And if an action potential occurs, then it will conduct, it'll move, basically like electricity in your home along electrical wires. They conduct electricity. The axon conducts this electrical signal, our action potential, all along the axon to the axon terminal. And the axon terminal, then that action potential tells the axon terminal to release neurotransmitter into the synapse, the chemical signals, and then that neurotransmitter binds to receptors on the postsynaptic cell membrane and generates another response in that cell. So basically, the processing of all that information is because of neurotransmitter binding to receptors and communicating to the next cell. So let's talk about that action potential. Remember, membrane potential is a separation of charge across the cell membrane. The membrane is creating a barrier, and the inside of the cell is more negative than the outside of the cell. So it's an unequal distribution of charge. And that unequal distribution of charge is basically called polarization. So the inside of the cell and versus the outside of the cell, they're polarized. And a depolarization changes the cell, the membrane potential to a more positive value. So if membrane potential is increasing from rest to some more positive value, that's called a depolarization. That depolarization occurs because neurotransmitter like acetylcholine, the abbreviation for acetylcholine is ACH, acetylcholine binds to receptors and opens channels that allow sodium to diffuse through into the cell. Remember, if sodium is allowed to diffuse across the membrane, the concentration of sodium outside the cell is higher than the inside of the cell. And if a channel opens that allows sodium to diffuse through, sodium will diffuse into the cell. And that positive ion, sodium ions, diffusing into the cell makes the inside of the cell more positive. Here it says less negative, same thing as more positive, or less polarized. So when the inside of the cell becomes more positive or less negative, that's called a depolarization. From that more positive value back to resting membrane potential. So if membrane potential is more positive and then it becomes more negative towards rest, that's called a repolarization. So when those sodium ion channels close and sodium's not allowed to move through the cell membrane anymore, but a potassium channel opens, and remember, potassium's high inside the cell. So if a channel that allows potassium to diffuse through opens, potassium will leave the cell. So as positive ions, potassium ions leave the cell, the inside of the cell becomes more negative. And as it becomes more negative, that's called repolarization. So here's what happens. Remember, sodium ions diffuse into the cell during depolarization. So if you're recording this graph now, is recording changes in membrane potential. So this blue line is resting membrane potential. It's negative, and this is just over time. So if sodium ions diffuse into the cell membrane depolarizes, becomes more positive or less negative. And as those channels close, that depolarization stops when sodium ion channels close and repolarization begins. And repolarization is due to potassium ions leaving the cell. And if positive potassium ions are leaving the cell membrane is changing, the membrane potential is changing from this positive value back towards a resting membrane potential that is more negative. So let's talk about how an action potential develops. There's two major phases, depolarization and repolarization. Graded membrane potentials depolarize the membrane to this value. And as the membrane depolarizes, it becomes more positive. So there's some sodium ions starting to diffuse through channels in the membrane. And that starts to increase membrane potential to some more positive value. Well, when this more positive value reaches a value called threshold potential, that is the membrane potential, the voltage at which an action potential is triggered. So a graded potential, remember I mentioned previously, graded potentials cause action potentials. So a graded membrane potential that depolarizes the cell membrane to a more positive value called threshold, it tends to be about 10 to 15 millivolts higher than rest. So if resting membrane potential, for example, is negative 70 millivolts, a threshold value may be negative 55 millivolts. That's more 10 to 15 millivolts higher than rest. That's the trigger for an action potential to occur. So the stimulus to generate an action potential is actually a graded potential depolarizing the cell membrane to threshold. And then at threshold, you get an action potential, generate an action potential. And then there's 3 steps involved in that. And we'll talk about those 3 steps, these 3 steps here on the next few slides. So actually, let me make sure I've got that on the next few slides. Maybe just this slide. Okay. So the 3 steps in an action potential are basically neurotransmitter has been released to the cell body that will generate an action potential. And that opens sodium channels. And that causes a depolarization. The depolarization, those sodium channels then close and potassium channels open, causing repolarization. And then the sodium basically back to resting membrane potential. So 2 of the major phases of an action potential are depolarization and repolarization. Threshold is reached and an action potential is triggered. At threshold, the neurotransmitter that has been released to cause the graded potential opens sodium channels, and then you get a huge depolarization. Those channels close, so no more sodium is able to diffuse into the cell, and potassium channels open, allowing potassium to leave the cell, diffuse out, and that causes repolarization back to resting membrane potential. And then once those ions have been diffused into sodium or out of potassium, the cell, the sodium-potassium pump basically pumps them back across the membrane to return that cell membrane to rest. So here's what happens. Sodium ion channels open, the cell depolarizes. Those channels then close. When these sodium-potassium ion channels open, that repolarizes the cell. And then now that sodium has diffused in, it needs to be pumped back out by way of the sodium-potassium pump. And when potassium gets pumped, it gets diffused out, it gets pumped back in by that same pump. So these 3 channels are all responsible for the generation of an action potential. The depolarization phase happens because of sodium ions diffusing in. Repolarization happens because of potassium ions diffusing out. And then to return the cell membrane to rest, the sodium-potassium pump pumps those ions back across the membrane using the energy from ATP. And there's a summary figure there to kind of summarize that. Those action potentials that get generated at the axon hillock of a neuron propagate. And propagate means conduct, basically, like electrical conduction of electricity on wires in your home. So continuous propagation is the mechanism that unmyelinated axons use to basically send an action potential from where it's generated at the axon hillock of a neuron, where the cell body meets the axon, all the way to the axon terminal, where it stimulates neurotransmitter release. So action potentials at one location of a membrane stimulate the next part of the membrane to generate another action potential. And that just continues down the axon. We'll talk about it's basically like dominoes falling and sending signals along the way, or from one to the next. Myelinated axons use this special type of propagation called saltatory propagation. Remember, myelin in axons, around axons, surrounds axons at certain points called internodes. The nodes are portions of the axon where there's no myelin. So basically what happens is action potentials only develop at nodes where there's no myelin. So you get this propagation that's faster because the myelin at the intranodes insulates that axon and allows the action potential to be propagated, conducted, sent from one node to the next node to the next node, instead of having to just kind of jump from part of the membrane to the next part of the membrane to the next part of the membrane. It skips the portions of the axon. The action potential skips the portions of the axon that are myelinated. So you send it faster along the axon. So here's what happens. Basically, where there's no myelin, one action potential propagates, is conducted to the next part of the membrane. So this action potential here, its job is to stimulate an action potential on this part of the membrane. And then the job of that action potential is to generate an action potential on the next part of the membrane, and then the next, and the next, and that will continue on and on and on and on until the action potential reaches the axon terminal, all the way down here. Well, that takes a while. So myelin, in this myelinated axon, basically allows this action potential to skip past the area where myelin's located and generate the next action potential in the axon in the node. So you get this basically continuous movement, conduction of the action potential from one area where it's generated, the axon hillock, to the next node of Ranvier, or node, to the next node, to the next node, to the next node. So basically, as opposed to just going from here to here to here to here really slowly, the action potential just conducts really quickly to the next node. So myelin insulates the axon so that the action potential doesn't have to propagate from down the entire axon, but it basically propagates from one node to the next node to the next node, all the way to the axon terminal. So it just happens a lot quicker. Less urgent information is sent along axons that are unmyelinated, but myelinated axons basically are located where information needs to be sent very quickly. And that information, that action potential information, is communicated from one neuron at the synaptic terminal, or the axon terminal, across the synapse. So I Googled this figure because I just really liked it. This figure shows you an example of one synapse. And I keep talking about that word, the synapse. It's made up of a presynaptic cell. And that presynaptic cell right here is the axon terminal of a presynaptic cell. And that presynaptic cell is one component of a synapse. This space here between the presynaptic cell before the synapse and the postsynaptic cell, some other cell, either another neuron or a muscle cell or a gland cell, the space between is called the synaptic cleft. So this cleft is a space. Neurons don't directly communicate to other tissues. There's a gap or a space between them, and that gap is called the synaptic cleft. And into that gap, neurons release neurotransmitter. So the job of the action potential that gets propagated from the axon hillock all down the axon to the axon terminal, its job, that action potential's job, is to stimulate the release of neurotransmitter. These little green balls are all neurotransmitter that's held in the axon terminal. And those neurotransmitters will be held in the axon terminal until the action potential says, okay, it's time to release. They release that neurotransmitter, it diffuses across the synaptic cleft, and right here on the postsynaptic cell membrane, there's a receptor to receive that, to allow that neurotransmitter to bind. And when that neurotransmitter, released from the presynaptic cell, binds to the receptor on the postsynaptic cell membrane, that then causes another response to communicate to the postsynaptic cell to say, here, you take this signal, and then you know what to do with it from here. There's a whole step-by-step process that we're going to talk about on this slide as to how that happens. So again, let's reiterate the action potential. So right here, this whole figure is showing you a synapse. This is the presynaptic cell, so this would be an axon terminal. And right, this red is the action potential, is propagating down the axon to the presynaptic cell membrane that's here. So this is the presynaptic cell membrane, the synaptic cleft, the space, and then the postsynaptic cell membrane where there's receptors for these neurotransmitters that will be eventually released. And this neurotransmitter happens to be acetylcholine. It's one of the major neurotransmitters. We'll talk about it here last before we finish up. So the action potential stimulates the release of neurotransmitter. neurotransmitter diffuses across the synaptic cleft and binds to receptors on the postsynaptic cell membrane. Those receptors happen to be sodium channels. And if there's sodium channels, when acetylcholine, the neurotransmitter, binds to those receptors, it opens those channels. It's the stimulus to open those channels and allow sodium to diffuse in. And if sodium is diffusing into the cell, the postsynaptic cell membrane is depolarizing. And that depolarizes the cell and generates an action potential that then stimulates that cell to respond. These neurotransmitters, acetylcholine is one I keep mentioning. Neurotransmitters are basically the chemical signal that allows neurons to communicate to any other cell in the body. And there's lots of different types of neurotransmitters, but acetylcholine is a major one. And we'll talk about acetylcholine a lot. So basically, acetylcholine has pretty widespread effects. It can be used by the central nervous system and the peripheral nervous system. Acetylcholine is typically the neurotransmitter that's used by somatic motor neurons to tell skeletal muscle to contract. So every time you contract your bicep or your tricep or your quadriceps, when you contract those muscles so that your body can move, there's a motor neuron releasing acetylcholine onto the membrane of that muscle cell, or all the muscle cells in that muscle, to tell them to contract. It's crazy, right? Norepinephrine and epinephrine are also neurotransmitters that generally have an excitatory effect. Serotonin and norepinephrine and epinephrine are used by the sympathetic nervous system. We'll talk about later. It's your fight or flight nervous system that responds and causes things like increases in heart rate, can control body temperature. So crazy things that these neurotransmitters can do. But basically, neurons have to release neurotransmitter to communicate to other cells. And what stimulates the release of that neurotransmitter is an action potential that we just talked about. Other neurotransmitters like serotonin, dopamine, glutamine, there's tons of different types of neurotransmitters. The major ones that we will talk about most are acetylcholine, maybe some epinephrine and norepinephrine we'll talk about later too. There's a review. So I will see you in the next slideshow. The next lecture is 7B and 7C. So we've talked about the basics of the nervous system now and how the nervous system communicates. We're going to get into some structures of the central nervous system, the brain and the spinal cord, and then a little bit about the peripheral nervous system too. So I will see you soon.